Hello, dear friends. We will continue with the lecture number 16 on cell and molecular biology. So, last time we have seen the nucleic acids, DNA and RNA. We have seen the structure of DNA, their base pairs, their nucleotides, nucleosides, then purines and pyrimidines, and everything we have seen. Again, we will uh, throw a look over these nucleosides plus phosphoric acid and the names of the nucleotides. We have seen the nucleosides. Now these are nucleotides. If we will add a phosphate sugar, it will form a nucleotide. Adenine plus deoxyribose sugar plus phosphate is adenylate acid. This is a nucleotide. Guanine deoxyribose sugar plus phosphate, guanylic acid. Cytosine plus deoxyribose sugar plus phosphate sugar, cytidilic acid, and thymine deoxyribose sugar plus phosphate is thymidilic acid. So these adenine, guanine, cytosine, and thymine. These are adenylate, guanylic, cytosinic, and thymidilic. So these all are the nucleotides formed by the union of these sugar and deoxyribose and phosphate group. Now, in the two polynucleotide chains of DNA, sugar and phosphate groups form the backbone of the DNA. So, they are the backbone of the DNA. While the nitrogenous bases like adenine, guanine, cytosine are present on the inner side of the double helix, and the purine of the one strand always pairs with the pyrimidine of the opposite strand, or vice versa. Always adenine pairs link with thymine and guanine pairs link with cytosine, means AT and CG is the pair. Thus A is complementary to T, means adenine is complementary to thymine, or vice versa, and G is complementary to C, or vice versa. A is linked with T with two weak non-covalent hydrogen bonds, that is A is equal to T, or double bonding to T, while C is linked with G with three non-covalent hydrogen bonds, that is C. Actually it should have one bond here, so these two bonds are very close, therefore they are not looking. Three bonds are here, three weak non-covalent bonds are here in cytosine and guanine. The nitrogenous bases are linked with pentose sugar. These are deoxyribose sugar and this is phosphate sugar. Here the pentose sugar by glycosidic bonds, while sugar and phosphate groups are linked by a phosphodiester bonds. Next, due to the constancy of the base pairing, the amount of adenine is equal to the amount of thymine, and the amount of guanine is equal to the amount of cytosine, because these are the pairs. A and T, adenine and thymine are pairs. Guanine and cytosine is a pair. AT and CG or GC alternate. Or we can say A plus G is equal to T plus C. This is the one side chain, this is the another side chain, and this is known as charge. This is known as Chargaff's equivalence rule. Chargaff's equivalence rule. The two polynucleotide chains of DNA are complementary to each other. That is, the sequence of bases of one chain is completely determined by the sequence of bases of other chain. For example, if the sequence of bases in one chain is 5' to 3' with AGATGC with 3', the sequence of the bases in the other chain is opposite, that is 3' to 5', and this chain will be reverse, that is TCA, TCTA, CG, because last G is pairing with C, G is pairing with C, A is pairing with T. In this way the chain is reverse here, and therefore the opposite chains are there on the DNA strand. In the double helix structure of DNA, one polynucleotide chain starts with 3 prime, that is 3 dash, and it ends with 5 prime, that is 5 dash end. So 3 dash and 5 dash, they are the two ends of the strand. 3 dash end refers to the third carbon atom of the pentose sugar, 3 dash end, which is free, while 5 dash end refers to the fifth carbon atom of the pentose sugar, which is free. These terminal These terminal 3 dash and 5 dash carbon atoms of pentose sugar are not linked to any nucleotide. The 3 dash end of one chain always lies opposite to 5 dash end of the other chain. So the two strands of the double helix DNA are called antiparallel end because they are opposite. In other words, the two chains of the DNA run anti-parallelly, that is, the two chains have opposite directions. That is, one chain has phosphate linkage to sugar groups is 3 dash 5 dash direction, while other chain has the phosphate linkage to sugar groups in 5 dash to 3 dash. So therefore these two chains are opposite to each other and therefore they are run anti-parallel, that is 5 dash to 3 dash direction. This is very simple. The O atoms of pentose sugar in one chain lies in upward direction and those of other chain lies in downward direction. The double helical structure of DNA can be compared to a ladder. In double strand DNA looks like a ladder, शिडी सारखा दिसतो म्हणून त्याला ladder शब्द वापरला आहे, where the two parallel sides of the ladder are made up of deoxyribose sugars and phosphoric acid molecules, and these are alternately present to form backbone of DNA. While the horizontal step of the ladder are made up of nitrogenous base pairs, the diameter of double helix is 20 degree angstrom and the distance between the two adjacent bases is 3.4 degree angstrom. As you have seen in the previous figure that 3.4 degree angstrom means 1 angstrom is equal to 1 upon 10,000 micron and 1 micron is equal to 1 upon 1000 millimeter. So this is the measurement of degree angstrom. Each base pair is rotated by 36 degree angle axis of the helix. Thus 10 base pairs make a complete turn of 360 degree angle. In other words, each turn of double helix covers a distance of 34 degree angstrom and 10 nucleotides are present in each turn. The twisting of the chains of DNA around each other forms a double helix which has narrow groove called minor groove and a wide groove called major groove. It has been suggested that the minor groove binds to some histone proteins and the major groove binds to non-histone proteins. I mean, they are not histone group proteins, just simple proteins are there. See different forms of DNA. We can see the types of DNA. The DNA double helix assumes different forms. Sugar occurring is the most important characteristic feature to distinguish the different forms of DNA. The observed structures of DNA depends on the nitrogenous base composition, salt content, and relative humidity of DNA sample. There are various forms of DNA, namely A, B, C, D, and Z DNA. These are the different forms of DNA. Some of these types or forms of DNA are discussed here. One, B form DNA or bDNA. B form DNA structure has been proposed by Watson and Crick. So these are the A form DNA, this is B form DNA, this is Z for DNA. If we will see from the frontal view, these are the different structures. A DNA somewhat compressed, B DNA somewhat relaxed and free, and Z DNA are compact. So we will discuss next. We will have a break here. We will concise today's lecture here, and we will take or we will continue this lecture next time. Sorry for inconvenience, but have a break here.